Hello, welcome to our lab. Today we'll perform a dissection of tomato plants. To start this protocol, you first need to wash your materials. In this case, we'll work with stems. After washing your tomato stems, you will perform fine cuts of this material in order to get as many slices as you can. Second, you will put these slices into a solution of toluidine. This chemical will react with the cells that are alive and will stain them blue. You will leave them for 1 minute in this solution. After the minute passes, you need to remove the excess of dye from the cells. You do this by putting the slices into a glass of water for 1 minute. After this minute, you need to repeat the process again, but with other chemical called fluoroglucinol. This one will bind to the cells that are dead and will stain them red. You need to leave these slices in the solution of fluoroglucinol for 1 minute. After this treatment with fluoroglucinol, you need to remove again the excess of dye by putting the slices in a glass of water for 1 minute. Finally, you need to observe the slices in order to analyze the structure of the tomato plant. You can do this by using a microscope. You will need to magnify by 100-fold in order to take good photos of the cells you already stained.
Now, I'm going to tell you some recommendations so your work in the laboratory is excellent. First, it is advisable to watch the section well to avoid contaminants and observing the under the microscope. Second, be very careful not to cut yourself and make fine cuts to make sample clear. 3. Make sure have many cuts in case some of them are damaged. 4. Leave alone for 1 minute, neither more nor less, so that they do not stay too strongly. 5. Do not wash the section too vigorously with water to avoid discoloration. And finally, lastly, use 100 x immersion oil.